Perfect. It's great to see everyone. I think I know just maybe about everyone on this call. It's a lot of familiar faces, so it's wonderful to chat with you all today. I'm going to be going through a little bit of what our lab has been working on roughly the last like 5, 6 years. There's a lot that's sort of in progress I'm not going to talk about, but some of the stuff that's relatively recently finished as well. And then if there's questions along the way, always happy to break off from the slides and have a nice discussion. So I'm going to talk sort of broadly about some of the things we've been working on in terms of determinants of anti-tumor immunity. And these are my disclosures. I think the only one that's really relevant for actually two of them that's probably relevant. The first one is a bit of a conflict, which is for Scholar Rock. I'm going to talk a little bit about their drug, which we've been testing in the lab and some of the data we have for that. They make a TGF-beta inhibitor, basically a TGF-beta trap for the latent form. And then really the Department of Defense, the CDMRP, as Brian had mentioned, a lot of this work, particularly some of the early work, started really out of support from the AKCI, for which I'm incredibly grateful. And so those are probably the two highlights to mention. And so, going back a little bit about the history of kidney cancer, we know that particularly clear cells is not something that's conventionally responsive to cytotoxic therapies. And one of the things that's always sort of interested me, well before I was specifically interested in kidney cancer, I was really interested in the idea of how the immune system could be used to control cancer. And RCC has really been a bit of a prototype for this. So going back now well over 100 years, there's all of these sort of anecdotes of kidney cancer being this immune responsive, immunogenic tumor. So this is a fun one just from well over 100 years ago from Charles H. Mayo, an observation about performing an nephrectomy in someone with pulmonary metastases, and the pulmonary metastases after the nephrectomy spontaneously regressed. Again, what is almost certainly an immune-mediated phenomenon. And this is not something that's limited to surgery. Over the years, over 50 years ago, this sort of phenomena has been described with radiation to one site and the rest of the metastases regress. And even in the modern era, in the absence of any other systemic therapy, there have been this sort of anecdotes of someone getting a COVID vaccine and again having regression of metastatic disease. Now, these are all obviously exceptions. These are very uncommon events. These are anecdotes, but it sort of highlights this idea that kidney cancer is sort of natively immune sensitive. And so with that, it's obviously no surprise that immunotherapy has really been a cornerstone of how this disease is treated for quite some time now, going back many years, many decades. That was really with cytokine therapy, things like interferon and IL-2. And I think we're all aware of the idea that IL-2 really did have this tremendous benefit for a limited number of patients, 5 to 7% of patients. And that was incredible, but we know that the vast majority did not do well, and that the median survival around that time from diagnosis till death was probably around 1 year on average. Fast forwarding to the targeted therapy era, the anti-angiogenic era, obviously help people live longer, which is wonderful. But really, none of those patients, maybe with very rare exceptions, were really cured of their disease and had meaningful, long-term outcomes with these TKIs. And I think that's true for just about every targeted therapy, again, with rare exceptions, that almost always the cancer will figure out a way to gain resistance and ultimately escape. A landmark moment happened in 2015 with this Checkmate 025 trial. This is really kind of the proof of concept that immune checkpoint inhibition can work in kidney cancer. And that's obviously been followed by the modern era that we live now in, which is ICIs plus TKIs or ICIs plus ICIs. And I think, again, I like to do the step through just because it's always, just amazing to me this incredible progress. We saw, the Brian Walpern present the RAS inhibition, and that was an incredible moment for pancreatic cancer in terms of this improvement in overall survival. And this has been a pretty remarkable change as well. We see sort of the median survival go from about 1 year to about 4 to 5 years. And I think the thing that makes me so enthusiastic about immune therapy specifically is the idea of this tail of the curve. And we see, this now extend out many, many years where there are some patients who are almost certainly cured of their disease. I don't, unfortunately, think it's as high as 30% like we see in clinical trials. I think that in reality, in the real world, it's probably a lower number, but there are some patients who I think really are cured of metastatic disease with immune therapy. But we also know that that's not true for most patients. And so I think we're at this moment now where it's this sort of proof of concept phase that curing a metastatic solid cancer, curing kidney cancer is a possibility, it's not a if it's doable, it's doable for some patients. And then how can we understand when it works, why it doesn't work in other patients to obviously try to make that more the norm. And so the simplified framework our lab has used for a number of years, this is starting to change, but I'll also mention this initial framework, which is a really kind of simplified view of the tumor in the microenvironment. And it looks like this, which is basically you have a kidney cancer cell interacting with T cell, and that obviously takes place in a complex and heterogeneous microenvironment with lots and lots of other cell types. And so even with this pretty simplified framework, we can ask some basic questions. What are the genetics of the tumor? What are the somatic alterations? And how do those impact things like the immune infiltration? And how do those things pair together to ultimately impact response or resistance to current therapies? What are the T cell populations that are there? What are the other immune cells that are there? And how do they talk to each other? What's the cell-cell communication? And then finally, we think really the heart of antigen-specific anti-tumor immunity is sort of shown here, where on the one side, you have tumors that present antigens on their MHC class 1 molecules, and that's recognized by cognate T cell receptors on CD8 T cells. And that's the interaction that allows T cells to ultimately find and kill tumor cells. And I would argue that for the most part, we don't really know either side of this equation very well. We don't know what are the target antigens when you have successful immunity, and we don't really know much about the T cells that recognize them. And so these are kind of the three big areas our lab has historically focused on, and that's what I'm going to talk about today is kind of stories from each of those three big buckets. I will say it doesn't escape me that this is a very localized view of kidney cancer. It's only what's happening in the microenvironment. And it's also only really what's happening at the cellular level. That's really what we're looking at. And so I think I'm not going to talk about it today, but the directions our lab has gone in recent years has tried to sort of expand a little, obviously continue along this light, but expand a little bit in a couple of ways. One is we know, for instance, that the cells themselves are floating in this metabolically lipid-filled, protein-full fluid, an interstitial fluid, that almost certainly impacts both T cell immunity and myeloid function. And we really haven't studied that much at all, largely because it's difficult to do. A lot of the techniques we have typically focused on analyzing the cells of the DNA. And so focusing on understanding the tumor interstitial fluid is something that we've developed a pretty big interest in its sort of immune modulating effect. Lots of great groups have begun to look at this already. Jeff Rathmel's group, Greg Delgoff's group, and others. So we have an ongoing work in that. And then looking beyond the sublocal environment, thinking regionally and systemically. So what is the draining lymph node doing in kidney cancer, both in mice, but more importantly, I think in people? How's that contributing to anti-tumor immunity? Is that really a reservoir of good sort of progenitor exhausted T cells? And then globally, what is the impact of having a tumor in place due to systemic immunity? Does it impair systemic immunity? When you remove a tumor, a primary tumor, does that actually restore you to kind of a immune homeostasis? These are all kind of questions that we're asking that sort of beyond the local. But for this talk, I'm really going to focus on this kind of model, this local model of kidney cancer. And we'll sort of step through one by one, starting with what are the genetics of kidney cancer and how do those sort of interact with T cell infiltration to ultimately impact survival or response. And so this work started actually supported by the AKCI. I think this was maybe one of the figures in my application to the AKCI a number of years ago. This was a wonderful partnership with BMS, where we were able to gain access to either their sequencing data or their tumors or both, from patients enrolled in the phase 1, phase 2, and phase 3 trial that led to the approval of the first immune checkpoint inhibitor in kidney cancer, nivolumab. And so really one of the first things we did was really just try to understand what is the genetic landscape of advanced kidney cancer. There had been really just like foundational work in understanding the genetics of kidney cancer in general from TracerX, from the TCGA, from others. Those often tend to have a skew towards earlier stage disease. And so one of the things that we think this work was maybe helpful for is honestly just providing a genetic landscape for advanced kidney cancer and how that might associate with clinical outcomes. And there wasn't a huge amount so necessarily new here. A lot of the familiar players, VHL, PBRM172, all of those are the most common here as well. All of these essentially, vast majority have 3P loss, 5Q gain. We do see an enrichment in certain things like loss of regions of 9P, and that's not surprising knowing the work from TracerX that sort of gains metastatic competency. And we do see some other things that we've sort of been interested in, like more deletions than we had typically seen before in regions of 6P, which contains a lot of antigen-presenting machinery. Again, raising the idea that maybe there's some loss of heterozygosity of some of these antigen-presenting molecules. But we asked some sort of initial questions just based on what was known about other tumor types. How do things like tumor mutation burden, neoantigen load, frameshift burden? This is from Sammer and Charlie Swanton's group had this hypothesis that high number of frameshifts was why kidney cancer was so immunogenic. And basically, none of these had any association with clinical benefit. And so none of these really showed any clear association with good outcomes with anti-PD-1. When we looked at individual genes that might, we found one, which was something that had been described by Ellie Van Allen, a loss of function mutation in PBRM1 was associated with improved outcomes with anti-PD-1. And we didn't observe this with mTOR inhibition, though, I think realistically, this is probably just indicating a favorable biology tumor. So I think I'm not incredibly convinced this is something that's incredibly unique to immune therapy specifically. I think these are basically better behaved tumors often when they have loss of function mutations in PBRM1, and that's what we sort of picked up in this analysis. So not a huge amount on the pure genetic front. And so then we next look to the immune microenvironment. And we tried to look at this in, again, the same lens that had been used in lung cancer and bladder cancer, this kind of classic lens of hot versus cold tumors, or highly CD8 infiltrated versus non-infiltrated or desert tumors. And then finally, a third type, which is this immune excluded, where CD8 T cells, the green dots, you can see marching up to the tumor margin, but unable to get into the tumor center to carry out effector function. And this has been described in many other tumor types, including work from Genentech, where in bladder cancer, maybe up to 50% in their cohorts had this immune exclusion phenotype, and it was associated with resistance to atezolizumab in that case. And what we found was not necessarily new, that these are highly immune infiltrated tumors, that very few of these are actually immune excluded. About a quarter of them or so are deserts, but the vast majority, about 3/4 of them, are immune infiltrated. But again, it wasn't as simple as just having a hot tumor led to better outcomes. We know obviously CD8 T cells are really critical, but just having them there did not seem to be the deciding factor, that regardless of whether you had an infiltrated tumor, an excluded tumor, a desert tumor, there weren't any very, very clear differences in clinical outcomes with anti-PD-1 therapy. So we didn't see too much looking at genetics alone. We didn't see too much looking at the immune microenvironment alone. So the next question we had is, are those two linked in some way? For instance, in infiltrated tumors, is there a different genetic landscape than these cold tumors? And so when we first looked at mutations, again, we looked mutation by mutation, and there was only one difference, and it was, again, this loss of function mutation in PBRM1. And what was perhaps a little surprising was that it was in these cold tumors, these immune deserts, that they were really heavily enriched for PBRM1 mutations, where nearly half of the immune deserts had mutations in PBRM1, whereas fewer than a quarter of them did for the immune infiltrated tumors. So these cold tumors were really enriched for these favorable PBRM1 mutations. When we looked at copy number changes, chromosomal changes, it was more complicated. They definitely seem to be infiltrated tumors seem to be more chromosomally unstable. They just had more copy number variants. And so what we did, shown here in this graph, is sort of look variant by variant and ask, okay, which of these are significantly increased in infiltrated tumors? And that's on the x-axis. And then within infiltrated tumors, which of them are significantly associated with clinical outcome? For instance, in this case, overall survival. And that's on the y-axis. And when we do that, there's really only one that rises to the top, deletions of 9p to 1.3. Again, this sort of locus that has CDKN2A, CDKN2B, MTAP, that has been associated with aggressive behavior. This was really associated with substantially worse outcomes, particularly in infiltrated tumors when treated with PD-1 therapy. And we can see that here for progression-free and overall survival, and it's just not something we observed for mTOR inhibition. And so, the kind of model we put forward is, this is kind of
David, can I interrupt a second?
Yeah, absolutely.
You just go back to the last slide. I hadn't really thought about this, having read this paper a long time ago. I mean, we know anti-PD-1 is better than mTOR all day, right? By 0.25 and just clinical experience. So I wonder if you're not seeing anything in mTOR just because all those patients do poorly. Do you know what I mean?
Oh,
that it's just prognostic and they do poorly.
I think that's absolutely right. I think my sort of view on this has come to be along those lines, that it's hard to say this is specific to immune therapy, but I think what's happening is essentially this. You have these infiltrated tumors that most cancers, you think these are poised to respond to anti-PD-1 therapy, and if they're cold, they wouldn't, but you basically have an enrichment in what are just clinically favorable, better biology tumors in this non-infiltrated group, and these are basically dragged down by being these sort of, more aggressive biology 9p21.3 deletions.
Just they have time to respond, right? PBRM1 is more indolent tumor, so they just have time to respond to immune therapy, whereas with mTOR therapy, they're not going to respond anyway.
Yeah, and there might truly be immune-mediated effects. I think Billy had shown some work about like PBRM1 basically amplifies HIF signaling, so maybe that upregulates endogenous retroviruses to some extent. And so there are arguments to be made that there might be some immune phenomenon, but I agree. I think it's hard to disentangle. And I think when we've looked at this in other kind of modern data sets in the first line, and the caveat being they're almost always in combination, we see it generally be favorable, but not necessarily discriminating between that and like a TKI outcome always.
Yeah. And last question, you don't have response, like objective response data, because that would cinch it, right? If tumors don't shrink on their own.
It did. So I didn't show here, but it was associated with better response with Nivo. Like the ORR was higher.
Okay. Thank you.
Yeah.
And so, David, can I follow up?
Yeah, please.
So the obviously the opposing cells can be immunosuppressive. I think, and you might get into this later, I suspect, with your myeloid type stories.
Yeah.
But early on, at least in bulk data, did you all look at whether Treg signatures or M2 like macrophages oppose this? In other words, were the infiltrated tumors just equally immune suppressed, and that explains sort of the conundrum you're describing?
We did. The hard part was we got to pretty small numbers, because by the time we sort of overlapped our Venn diagrams of where we had IF data to define infiltrated tumors, where we had exome data to define the copy number loss, and then when we had RNA data to do the immune convolution, that intersection is pretty small. And so we didn't see any like major immune population. We did immune deconvolution and all kind of the standard stuff, and we just didn't see anything that would clearly sort of jumped out as the rationale.
All right, sounds good.
Abhishek, I think you had a question.
It was more of a thought, David, and this was in line with even some of the previous ideas as well, right? So the cohorts that we are comparing probably are not identical, perhaps, with the immunotherapy and the mTOR inhibitor being used, right? So they're in probably the mTOR inhibitor is going like tumor refractory to almost everything else before that, or were they kind of randomized at the same time?
Oh yeah, so this is a randomized trial. So this is like patient comes in and they're randomized to get one or the other. So
I see. So they're essentially seeing the tumors with the same initial, to whatever degree it can be controlled, a similar genetic profile. The
Exactly.
second aspect really to think about, and this might not be completely aligned with what your long-term goals are, but there's something that at least I am thinking a lot about is, and maybe you should be thinking with the metabolic ideas that you were throwing out, is the impact of the metabolic perturbation, including mTOR inhibition, which I think of as a metabolic perturbation essentially in the tumor. The impact on the tumor intrinsic oncogenic program and the immune cells is often not going in the same direction that you want to,
Sure.
right? So you can have a beneficial anti-metabolite on the tumor intrinsic pathways, but that might also at the same time come with a collateral damage of inactivating the immune cells also. So I think this is a part that I think that we need to start thinking a lot more aggressively about, including in the experiment that have, not the experiment, or the clinical trial where mTOR inhibitor has been used. Where did we leave the immune cells in the tumor? What state were they left behind in? Is a question that I don't think we have sufficiently answered. I can tell you in some of our preclinical ex vivo studies, we hit tumor cells in the presence of PBMCs with these metabolites, and the T cells become completely exhausted in the face of these inhibitors. So it's just an idea to think about.
No, I think it's a really important thing, especially as we start to think about combination therapies. I'm not going to talk about it here, but we actually have a paper in revision at JCI now about basically the impact of belzutifan on T cells. I think Katie Becker had mentioned it during her ASCO discussion, ASCO GU discussion, but it's, again, this idea that, are these therapies, maybe they're beneficial on the tumor intrinsic side, but are they actually damaging on an immune side? So, obviously what we're hoping for immune therapy, and again, I think somewhat distinctly from other forms of cancer therapies, is really this idea of exceptional response, that anti-PD-1 therapy really has the potential to lead to long-lasting, durable responses, even hopefully cures for some patients. And so we really began to study, are there sort of defining features of those exceptional responses? And so to do that, we sort of made up what is, in all honesty, I think reasonably clinically defensible, though somewhat arbitrary definition of what an exceptional response would be, which is a complete response lasting at least a year, a deep partial response that's lasting at least 2 years, or a partial response at all in immune therapy lasting 3 years. And I think we know from some of these studies, if you're still in response for 3 years on immune therapy, you're likely to kind of do well in the longer term. And so we were able to, again, work with BMS to get access to the Checkmate 214 data, and with Pfizer for the Javelin Renal 101 data, and sort of define these categories of exceptional response, intermediate response, which is sort of response that doesn't meet that our exceptional category, and then the other stable disease and progressive disease. I will just sort of make one note here, slightly discouragingly, but perhaps not surprisingly, that if we look at the green bar for exceptional responders under a Checkmate 214 trial versus our IMDC, it's just, we do see a lot higher proportion of exceptional responses in the trial setting than in the IMDC kind of real world setting. And I think that's probably reflects a lot of people's experience as well. It's not that we don't have exceptional responses. I think it is probably higher than we see in the real world in the Checkmate 214.
David, why is it not different for IOTKI?
Yeah, it's
like
a
if
good
it was just trial bias and patient selection, you'd think that would apply equally.
Yeah,
it's a real mismatch there, though.
Yeah, I will say, if anything, and I obviously am an immune enthusiast, if anything, it's actually the opposite trend a little bit for the IOTKI, because these are absolute numbers, but you can see the total size of the bar for IMDC for IOTKI at that time was smaller. And so it actually, like, the relative proportion that were exceptional responders was probably even slightly higher in the real world than was in Javelin Renal 101.
And David, is the trial here Javelin, so it's PDL1 and your IMDC?
Yeah.
Probably
I think
has
that's probably
PD-1.
Yeah, I think that's actually, Steve, a very good point. It's because I think what we did here was the trial was avelumab, and then for IMDC we just took any IOTKI. And so I think that is probably a little bit inflated, because probably is more exceptional responses to Nivo, Cabo, Pemberlen.
Gotcha.
Perfect. And then, one of our interests certainly was in investigating neoantigens. We had begun to do that at the beginning, but I want to do a little bit more in depth here. And just to get everyone on the same page, I think just about everyone is familiar, but because I'm going to come back to neoantigens at the end, typically all the proteins in our intracellular proteins get degraded, chopped up, loaded and presented onto MHC class 1 molecules, and that's our natural surveillance mechanism, and T cells know to not recognize self in general and don't respond to them. But if you have a non-synonymous mutation that leads to an amino acid change at the protein level, that could be a driver mutation or it could be a passenger mutation, just something that randomly happens that doesn't actually contribute to oncogenesis. That will now be a new peptide that the immune system, the T cells, have hopefully not been educated to ignore. There's not tolerance to it. And so then T cells should be available to recognize and attack those tumor cells. And so in other tumor types, we know that we use TMB essentially as a surrogate for neoantigens because neoantigens are sort of more complicated to assess and predict. But we do have fairly good tools now to be able to predict them. And what we found here was that it's not that neoantigens don't matter, which is what I think was our initial inclination from our first studies and from a lot of studies that sort of show this negative association with TMB, that we never see this or the no association with TMB. We sort of assume that maybe neoantigens don't make a difference. I don't think that's true. That if we look at really those patients who have an exceptional response to immunotherapy, and we look at not all neoantigens, but particularly clonal neoantigens. So these are neoantigens derived from mutations that are truncal, so like the ones you'd have in VHL or PBRM1 or BAP1, the ones that are present in every tumor cell, those are really, this is a log scale, so those are really highly enriched in exceptional responders compared to either those that don't respond or intermediate responders associated with substantially better outcomes too, even if we try to control for a lot of other factors. And so we really think clonal neoantigens are really an important factor for those patients who have exceptional responses.
So David, I have a conceptual question over here, and this is something that I think it's in line with some of the topics that I've heard about. How long should patients be on therapy if they're responding? Once the initial response is occurring and the T cell has recognized the neoantigen and cleared out the tumor with the truncal mutation, the antigen is probably gone, right? The stronger the immunological response, conceptually, you can be nearing a state where the antigen is now down to 0 in the host. In that state now, is the immunological regulator actually achieving any beneficial effect against the tumor because the T cells are there, but there's no antigen to recognize and clear out anymore?
So almost certainly now, at that point, you would typically diminish your T cell pool and you'd maintain like a memory reservoir. Like that's the same thing that would happen in a viral infection. But I think the reality is you rarely get to that true tumor zero, right? That to very, I think the answer would almost be sort of obvious that if you truly got to tumor zero, you definitely wouldn't need immune therapy because you wouldn't have any residual tumor cells. But I think what almost certainly happens is, right, you get to some minimal level that if you think about like the 3 E's of immunoediting, right? We're at that last phase of immune escape. Maybe you sort of are pushing it back into that middle phase of immune equilibrium.
And just a very quick follow-up on the earlier question. Has there been a situation where we have compared anti-PD-1 and anti-PD-L1 in a randomized situation?
No, I don't think that would.
So our assumption that they are not equal is based on unrelated cohorts being compared in some way.
Yeah,
It's just
Prajay. I think the
I was
overall
say just multiple phase 3s.
Yeah.
Right. Not comparative, but I think there's 5,000 patients randomized. I think you get a sense.
Yeah, like if you look at all of the IO anti-angiogenic things, and obviously there's flaws here too, but Checkmate 9ER has a survival benefit. Keno 426 has a survival benefit. Clear has a overall survival benefit. Emotion 151 does not. Javelin Renal 101 does not have an overall survival benefit. And so I think every time you use a anti-PD-1, you have an overall survival benefit. Every time you use anti-PD-L1, you don't. So obviously that's not proof, but I think it sort of supports this clinical gestalt. It is clinically active. Like I think from David McDermott's Emotion 150, they had a single arm. They had an arm that was single agent atezo that clearly has clinical activity. So it's not like an inert thing, but it's not, I think that aggregate data sort of suggests it's not the same. Great. And so if we now turn to our transcriptional data and ask again, similar question, this is in the IO-TKI cohort, what is different between exceptional responders and progressive disease? We see a lot of immune pathways come up, but perhaps maybe shouldn't have been surprising, was a little surprising to us. They were mostly B-cell pathways. And when we do our sort of standard immune deconvolution, what we see is certainly CD8 T cells are enriched in exceptional responders, but together with plasma cells and B cells. And this immediately got us thinking about the idea of lymphoid aggregates or tertiary lymphoid structures. I think there's a lot of debate about exactly the nomenclature for these things. In mice, it's clear that at first they're just aggregates of lymphoid cells, and then eventually they might progress to being immature tertiary lymphoid structures, and then eventually can progress to being mature tertiary lymphoid structures that have germinal centers and HEVs and these sorts of things. I'm not sure the human, at least from what we've seen in RC, the human sort of necessarily mirrors that perfectly. But these lymphoid aggregates clearly were associated with much, much better outcomes. We've done this both by immunofluorescence, so with Sabina Signoretti, and also transcriptionally using a number of signatures that these are clearly enriched in exceptional response. And so I'll skip ahead just to the last part, which is a little bit of ongoing work that is really hoping to be a resource paper more than anything else. This is together with Tony and with Ellie, which was an attempt to basically put together all of our cohorts, analyze them in a harmonized way, and provide that publicly with all the clinical annotations so that everyone can sort of use this sort of data to ask these sorts of questions. And so a lot of this was motivated by the fact that despite these studies seeming to be decently powered, they're actually dramatically underpowered for what we want to try to find. So what we were trying to do is essentially find individual genes that might be enriched in responders, for instance, compared to non-responders. And our initial study, which I think had 450 patients or something around there, seemed like it was a large number. But actually, when we do the power calculations for the number of patients treated with checkpoint inhibition, we were actually powered to find basically the one thing we found. We were powered to find high-frequency mutations that are really enriched in responders compared to non-responders. And so what we've done is put together a much larger cohort of about 2,000 patients by aggregating all of these together. And now we're powered to really detect a lot more sort of low-frequency mutations that might be present at 1% or 2%. And so this is a giant plot of what that sort of looks like. This is 2,000 patients with whole exome RNA-seq data and clinical annotations. And we really sort of get a sense of the genetic landscape. This is not in this figure, but there really is a very long tail of recurrent mutations of about 100 or so driver mutations that we think are at least recurrent mutations, significantly recurrent mutations that are higher than we'd expect by chance for this cohort. And so with this, we can begin to look at things that are lower frequency, like NF2 mutations, and start to understand, okay, what are the genetic associations with NF2 mutations? These are, I'll say, not like BHP tumors. These are true clear cell tumors. And how do these things associate with clinical outcomes? So this is something that took a long time just to not particularly honestly just like the technical element of like harmonizing these data sets, analyzing in the same way, but we're hoping to kind of get this out within the next number of months and just have this as kind of a resource paper. So with that, the first part of this, I'm probably going to speed through the second part, is really about understanding the genetics of kidney cancer. I think one of the things we're really interested in the future is, again, looking at a larger scale and then ultimately integrating some machine learning-based approaches to help predict clinical outcome. David Van Dyke is a really wonderful computational, I was going to say computational biologist, he's just a computer scientist here at Yale. He developed a foundation model for single cell data called Cell to Sentence that he developed with Google. And so we are working to adapt that cell to sentence from a single cell framework to a bulk framework to try to see, can we use these foundation models to really try to better predict clinical outcomes. So the next question is really about the tumor microenvironment. And I'm going to, in the interest of time, skip through part of this, but it was really motivated by what are the cells that are present and how do they change in different disease conditions. And so we started off looking at the RCC baseline, meaning an unperturbed setting, what the immune microenvironment looks like, and then more recently have begun to look at determinants of response resistance. I'm probably going to skip over this first study because it was published a number of years ago during my AKCI period, and I'll skip to kind of the summary, which is, we think that there is a sort of evolution of the immune system as you advance along disease stages. That when you start off with early stage disease, there's fewer exhausted CD8 T cells that are exhausted. They have a more functional state. The macrophages have a more pro-inflammatory phenotype. And then as you move to locally advanced and ultimately metastatic disease, both the T cell and the myeloid phenotypes change, but they're not independent, that they really seem like they're talking to one another and probably supporting one another in a way that we think is supporting the sort of immune dysfunction. More recently, we've been interested in again, doing this sort of immune landscape analysis in non-clear cell types, but then also looking at how this might impact therapeutic response and resistance. And so first of all, looking at non-clear cell types, this was a large effort really done together with close collaboration with Lisa Henske's group for a number of years, where we looked at chromophobe kidney cancer and a number of other oncocytic tumors. On the one side, you have oncocytomas, which are classically benign, low-grade oncocytic tumors, which are debatably benign versus malignant, but have a good prognosis, and then chromophobe RCC, and began to ask a series of questions, including can we confirm cell of origin, but also really what does the immune landscape look like? And so, like many others before us, we looked at just the total amount of immune infiltration, and unsurprisingly, the total immune infiltration in these oncocytic tumors is very, very low compared to clear cell kidney cancer, so really low immune infiltration. Of the cells, immune cells that are there, very few of them are CD8 T cells. So by single cell analysis, we can say what is the relative proportion of different immune populations. In the darker colors is chromophobe, the lighter colors is clear cell. The relative proportion of CD8 T cells is only around 10% or less of the total immune population, which is already low. So we have a tiny, tiny number of absolute number of CD8 T cells getting into chromophobe. Out of the ones that are there, the question is, are they any good? Are they even capable of recognizing tumor, or are they just bystanders? And so to look at this, we used a single cell signature of T cells that are tumor specific or capable of reacting to tumor versus those that are bystanders or viral reactive. And again, unfortunately, here we see that for clear cell, most of the cells that are there have a high tumor specificity signature, indicating that in large part they're capable of recognizing tumor compared to their viral specificity. But we see the opposite pattern for chromophobe, that most of those have low tumor specificity and high viral specificity. The thought is they're likely bystanders. We used an approach that was originally developed by Sarah Teichmann's group to try to identify the cell of origin for chromophobe kidney cancer and confirm what other groups had seen, that this looks like it comes from the alpha intercalated cell. But then what we could do with the single cell data is really compare how the tumor cell itself is different transcriptionally from its cell of origin. So often when you do bulk RNA-seq, you might pick up a bunch of things that are upregulated, but that's just reflective of it being from an alpha intercalated cell, and alpha intercalated cells have those same genes upregulated. So this gives us a window to really how it's different from its cell of origin. And we see certain things, that pathways that are increased. Lisa has been very interested in ferroptosis in chromophobe. We see certainly mTOR signaling increased in these tumors. But then we also see a number of things downregulated. And the one that really stood out to us was antigen processing and presentation. We see clear downregulation of HLA class 1 molecules. I will mention as well work from MSK that's looked at IL-15, particularly as sort of a booster for ILC1s in chromophobe. And Martin Voss has been trying to treat a few patients on single patient INDs with IL-15. And so how does this actually correspond to clinical outcomes? And again, huge caveats here. This is working with Danny Hang again with IMDC. These are first-line patients, but there's I'll sort of mention the caveats as I go. But we try to get a sense of, for first-line patients treated with a TKI versus ICI versus mTOR, how do they do between chromophobe and clear cell? And we can see that for TKIs, on average, there's lots and lots of patients. On average, those lines are reasonably overlapping, maybe subtle differences, but reasonably overlapping. For immune checkpoint inhibition, they do substantially worse with chromophobe RCC. And then for mTOR inhibition, we can see that they actually do substantially better with chromophobe when they're treated with mTOR inhibition. The giant caveat I'll give there is, obviously this is real world, so we have to think about who are the patients with clear cell that are being treated with an mTOR inhibitor in the first line, which is probably a select group of people treated with temsirolimus a number of years ago. So that's the big caveat here. But it's also how we can just clearly see in absolute terms that the patients with chromophobe have a small numbers, but median OS of 41 months when they're treated with an mTOR-based regimen first. So I think in general sort of supports this idea that a lot of these might be mTOR-sensitive tumors. And so, on the immune side, which is what we're interested in, we really do think these are just totally different beasts than clear cell, and so are going to need just a dramatically different approach immunologically, whether that's IL-15 to engage ILC1s. I think it'd be really interesting to think about T-cell engager therapies to actually bring in T-cells that you don't necessarily care if they're tumor reactive, but you can bring them in and activate them. I think there's going to be just new approaches that are needed for chromophobe. And so finally, I'll sort of end this section just thinking about how we can apply this to understanding response and resistance. And I think this is more commonly done now. I think when we started to think about this a number of years ago, it was not immediately clear how to do this, because it's actually collecting samples in a way that you could cryopreserve them for single cell at 10 different sites across the country and have that work was challenging. Now there's new technologies that maybe make that easier. But at the time, we sort of had this idea that maybe we could do this for a select few patients, that within a clinical trial setting, we could build in a kind of a discovery cohort where we would collect this detailed information and do this more in-depth analysis, generate hypotheses, and then use the remaining part of that clinical trial, the remaining patients, as sort of the validation using much more conventional means. And so our sort of first attempt to do this was really building into Mike Atkins's HCRN-GU 16260 trial, which was a really nice clinical trial, answering a really good clinical question, which was, can you give anti-PD-1 monotherapy and salvage with IPI later? But for our purposes, it was just a trial of anti-PD-1 monotherapy as a first-line therapy for kidney cancer, where we could sort of test this model of, for a select number of patients, I think 17 patients, we were able to collect single-cell RNA-seq, and then for a vast majority more, get kind of classic whole exome sequencing, RNA sequencing, immunofluorescence. And so we performed single cell RNA sequencing, focusing on the T cells. Again, we see lots of different T cell populations. And so we performed a kind of similar analysis as before. We asked which T cell population is significantly associated with therapeutic response. That's on the x-axis. Actually, that could be positively or negatively, is associated with response or resistance. And then which of those populations are significantly associated with altered progression-free survival, and that's on the y-axis. And there's really only one population that came up. It was a CD8 population that expressed lots of markers, classic expression markers for T-cell exhaustion, like TOX, lots of co-inhibitory receptors. Also expressed a lot of genes that are associated with a tissue residency program. I've highlighted Hobbit here, ZNS683, but CD69, CD103, a lot of things we think of with tissue residency. And this homotypic receptor that I don't typically think of as a T cell thing, SLAMF7. It's really high on plasma cells, on NK cells, but we saw it here as well. And so this population was associated with really bad outcomes in this small number of patients. We see the proportion really increased in progressive disease, and a high proportion was associated with significantly worse progression-free survival. And so then we made a gene expression signature that represents this population and asked, okay, beyond these 13 patients, does it look like it's associated with altered outcomes in the rest of the HCRN cohort, which is at the top, or in our original Checkmate 025 cohort? And again, it was associated with significantly worse outcomes with progressive disease and significantly worse PFS. The question always is this just like a a biomarker? Is this a marker of a bad immune state, or is this somehow like a functional impact? Is for instance SLAMF7 decreasing T cell function? Is this leading to worse responses? And so we've begun to test that in a couple of different ways. For this first paper, we really just looked at SLAMF7. So we expressed SLAMF7 on healthy donor T cells. It's a homotypic receptor, so we expressed it also on K562, so it provided the ligand for it. And we asked, does SLAMF7 just lead to any decrement in T cell function, looking at things like proliferation or cytokine production or markers of cytotoxicity? And the short answer was yes, that SLAMF7 led to a decrease in T cell proliferation, both CD4s and CD8s, decrease in effector cytokine production like interferon gamma, and then we've done more numbers since then, decrease in cytotoxicity. So we think this is actually having a negative effect on T cell function itself. We since then have really expanded this analysis to ask questions about what's causing this population, and is that sort of something we can intervene on? And so this is more recent work from Soki and Zhaochen and Rishab in the lab, where we have a much larger data set now of about 70 separate tumors that have been treated with immune checkpoint inhibitors that we've done single cell analysis for. And again, what we've tried to do is infer what are the soluble factors that might be leading to this tissue residency program in CD8 T cells. And that's what this niche analysis is showing here. And there's one that really sort of jumped out at us, which was TGF-beta, that really TGF-beta was sort of inferred computationally to really be leading to this tissue residency program. And so we want to see, in addition, at least, this TGF-beta really drive the phenotype and the dysfunction that we're seeing. And so in hypoxic conditions, we can take healthy donor T cells, we can treat them with TGF-beta, and we really do recover this immunophenotype, this tissue residency exhausted population that expresses PD-1 and TIM3, but also tissue residency markers CD103 and CD69. It directly impairs markers of T cell function. We've done a couple of different versions of this. This is just the cytokines it can produce, like interferon gamma, are decreased with TGF-beta, but it also directly impairs T cells' ability to kill tumor. And so what we have here in this system is we've expressed a model antigen, NY-ESO-1, on our tumor cells, and then we've transduced healthy donor T cells with a T cell receptor that recognizes NY-ESO-1. And so as you increase your T cells, you will kill tumor cells. But if we add TGF-beta, it essentially inhibits the T cells from killing tumor cells. And so we really think that this is in part leading to this dysfunction, the TGF-beta production by a variety of cell types, including Tregs. To Billy's point earlier, I think that's probably one of the main cell types that's producing TGF-beta. And so the question is this potentially reversible? And so TGF-beta is made in a latent form, and then it needs to be activated. And there's a bunch of activators that are available that can then cleave this and basically release TGF-beta to its active form. And so what we wanted to see is, okay, using something that's actually clinically available, it's now been published, that's actually had some activity in kidney cancer, is this actually able to restore T cell function in some way? And so the setup for this assay is we take healthy donor T cells, we stimulate them in the presence of this latent TGF-beta that's plate bound. We can add the activator or not, and then we can also add our drug or control antibody. And so we if we take our T cells and stimulate them in the presence of just the latent form of TGF-beta that's inactive, it doesn't do anything, and they make lots and lots of interferon gamma. If we add the activator, so now it's active TGF-beta in this system, we see inhibition of interferon gamma production. A control antibody and isotype doesn't do anything, but then if we add the Scholar Rock drug, now we can restore T cell function, at least here measured by interferon gamma. And so we think that really this sort of TGF-beta leading to this sort of tissue residency program and impaired function is really at least one cause of resistance within RCC, and hopefully something that's sort of clinically actionable. So I know there's only a few minutes left, so I'll maybe just quickly run through the last part, which is a huge interest of ours, is this last part, which is understanding what are the antigens that are relevant in kidney cancer and how we might therapeutically target them. And so to test really this hypothesis directly in people, this was a multi-year effort, but we launched a phase 1 neoantigen vaccine in patients with high risk but resectable kidney cancer, so stage 3 or stage 4, but with oligometastatic disease, so we could fully resect it. We'd predict antigens, synthesize, in this case, synthetic long peptides for up to 20 neoantigens, and deliver them in a somewhat complicated 4-pool system in this prime boost format. And so there are a number of questions. The first is this just feasible? Is this doable for a low mutation burden tumor like kidney cancer? It's not melanoma, it's not lung cancer, bladder cancer. It has very modest number of mutations. So for all 9 patients, despite having a very modest mutation burden, we could create a multi-epitope vaccine for all of these patients. We could vaccinate against single nucleotide variants, against frame-shift indels, against driver mutations. So it's doable. In terms of safety, it was reasonably favorable. I think just about everyone got 1 to 2 days of flu-like symptoms with each of these injections. So it's not nothing, but it's also time limited. There's only 7 injections, and so nothing that lasted longer than that. And then everyone had an injection site reaction at the site of the injections, but again, that was transient. The next question was, does it lead to actually an immune response? And the short answer is yes, it does for all patients. This is a summary of a lot of interferon gamma ELISpot data where we basically take the patient's T cells out at various time points before vaccination, during, and after. We add our peptides ex vivo and see do the T cells recognize them. And so each row is a patient and their 4 peptide pools. Each column is a time point. So if we start all the way on the left at time point 0, we see it's basically all white, for the most part, indicating we can't really detect pre-existing T cells that are capable of recognizing these neoantigens that are circulating. Doesn't mean they're not there. If they're there, they're below the limit of detection. But for every single patient, we see a detectable level of T cells that are neoantigen reactive. If we look at some of the ones that elicited what I would call the best or strongest immune responses, it really was against these clonal neoantigens that we sort of found associated with exceptional responses earlier. And we can see in the prototype here for VHL, where each of these dots is 1 T cell that's reacting, and really a high level response against VHL, but that was true for a number of clonal driver mutations. I will say that's for reasons we don't fully understand. I wouldn't think a priori that the type of mutation would really have any impact whatsoever on whether something leads to an immune response, but just definitely an observation we had. The next question is this immune response actually durable? To do this, it was actually a little bit complicated, but we had to track T cells that were vaccine reactive over time. And so the way we did that is, people might remember the old tuberculosis tests, the PBD, where you would inject a small amount of peptides from MTB into the skin, and if it blew up, that means you were reactive. That means you had a T cell memory against it. So we basically did that here. We injected a small amount of all 20 peptides into the forearm. We waited 2 days. They got a little skin bump. We would biopsy that, take out the T cells, and then single cell sequence those T cells so that we could see those are the vaccine reactive T cells. And now that we had those TCRs that were reactive, we could track them over time. So we could use just bulk TCR sequencing to track the TCR levels for vaccine reactive T cells. And what we can see is they're basically undetectable prior to vaccination. They shoot up, and they basically persist. They dip a little bit, but they persist well after the vaccine has finished. So it really leads to durable T cell memory. And then what we then did was take those vaccine reactive T cells and actually put them on top of autologous tumor cells. So we had kept autologous tumor cryopreserved for each patient, and we could test whether they were actually capable of reacting against their own tumor. And it wasn't 100% of patients, but for 7 of 9 patients, we could detect that the vaccine reactive T cells were capable of recognizing their own tumor cells. The big question is how do they do clinically? And the real answer is we don't know because these are 9 patients. Thankfully, they all did well, and that's really encouraging. But these are 9 patients that could have just done well with surgery alone, so we don't truly know. But it was encouraging. There were reasonably high-risk patients that they did well, and this helped to provide at least some of the motivation for this INTERPATH 004 study, which is now fully accrued, which is the randomized trial of a Moderna version of this, which is a RNA-based neoantigen vaccine. I'll end off just by saying that neoantigens are not the only drivers of immunity. We think there's lots of other antigen sources as well. One that's really been led by Chin-Chin Zhang in Bill Kaelin's lab really helped identify endogenous retroviruses as one additional potential source of antigens that can lead to T cell responses both in patients and also potentially as a vaccine target. These are in HLA transgenic mice. And so with that, I'll just maybe end here with lots and lots of people to thank. The folks in my lab who really did the vast majority of this work, close collaborators in many different domains, our funding sources, my Yale collaborators, the patients and their families that enable this work, our funders, and just a particular shout out to the AKCI, which is really what enabled all of this work to start. And with that,
Thanks.
1 minute left for questions.
All right, we have 30 seconds for questions. No, thanks, David. That was great. Congrats on all your work over the years. I'm going to have to jump off to lead another meeting, but I think I can leave it open and people can ask questions as long as David can stay.
Perfect. I do have a 1 p.m. meeting, but happy to chat. Oh yeah, well, go ahead.
Yeah, quick question, David. How are you thinking about the microbiome interacting with some of these, especially the tumor microenvironment, call it metabolite, call it, liquid biopsy, et cetera, since it appears that may be the mechanism by which the microbiome may have an impact, or at least one of the possible mechanisms?
Yeah, it's a great question. It's obviously incredibly complicated, but I think that you're 100% right that I suspect the link between them is that whatever favorable thing in the microbiome is producing some metabolite that's then leading to some favorable T cell change. And I think we see that, we don't know, for instance, for CBM-588, obviously, will it be effective? But if it is, how it's doing it, one naive thing is it's Clostridium butyricum, just it produces butyrate, and it's butyrate we know is sort of helpful for T cell function. And so I think making that link is something that I think is probably the next. The first step is just, it doesn't work, does it help? And I think there's more and more things between the work from Laurent Sivogel and the MADCAM1 sort of serum surrogate for the tissue level MADCAM1. I think there's more and more evidence of this idea of like the favorable gut microbiome versus dysbiosis and how that might contribute. But I think for me, the interesting thing would really be finding the link of what's actually causing it, because I think we really don't know. I think the last thing I'll mention is just relate to some of actually Steve's work on exercise. The group at Pitt, for instance, has also found exercise really engages the gut microbiome to produce more formic acid. And again, that's helpful for, at least in mice, helpful for T cell function. And so I think probably the missing piece in a lot of these is what that actual link is between the gut microbiome and that kind of factor X that then leads to an improved immune function. I apologize to hop off, but thank you, everyone.